डॉक्टर अर्जुन सूर्या एंड उनके बारे में आपको बताना चाहूँगा. डॉक्टर सूर्या हैज़ मोर देन 30 years of research experience and has held leadership positions of increasing seniority in the industry. He founded Curadev and in less than a decade, Curadev has emerged as one of India's rare drug discovery success story with a slew of first-in-class small molecule patents that have emerged from its unique approach to target selection and drug discovery. So I would like to call Dr. Arjun Surya.
I'd like to thank the organizers for giving us this opportunity. Am I audible? You can hear me? So today I'm just going to tell you a little bit about our lead program. That's a systemic sting agonist for solid tumors. Very quickly, a little bit about the company. We're a clinical stage biotech company. Started in 2010 in India, and now we have offices in the UK and the US. We're focused essentially on first-in-class targets. So we don't take anything that's, when you start a program, there's nothing in the clinic. So we don't make the next EGFR or the next, next or improved kind of compound. We work on things which are early stage, very early stage. So there's nothing in the clinic when we start a program. It's a very small group. We're about total of 10 senior people and total of about 50 scientists who are working in this space. The clinical development arm was established just 2 years ago because our first compound is ready for clinical trials, and so we decided to start building that up, and that trial started at Sloan Kettering. Very quickly, this is just a short history of the company. We started in 2010. Main key issues, the key, this doesn't really point very well. So 2015 we out-licensed our first molecules to Roche, which was a very big deal for us because it's not working, right? This thing?
Pointer काम नहीं कर रहा, इसका pointer काम नहीं कर रहा.
Okay, it's okay. I'll just say it. We licensed our first molecule, which is an IDO-TDO inhibitor, to Roche in 2015, and then sting agonist ADC to Takeda in 2019, and then a antagonist program was licensed by Bayer in 2020. So the sting agonist get taken systemically forward for cancer. We essentially have a whole bunch of programs but today what I'm going to talk about is a sting activator program. As a company, we are very sort of academically focused, so we have established relationships with many different organizations, including Tata Memorial, CMC Vellore in India, as well as organizations outside. So I'm just going to focus on the cancer immunity cycle. This is our real focus has been on this, and if you look at the problem, you can divide it into parts of the cancer's immunomodulation, in which you have cancers which are called immune deserts, where you don't have any immune system recognition of the tumor at all. You have cancers where you have immune exhaustion, and you can say cancers where there's immune suppression, and cancers where there's hypoxia, which essentially different parts of the tumor are so hypoxic that you actually have epigenetic modification occurring in those areas. So we've sort of picked targets in each of these spaces. The one that's for the immune deserts is the allosteric intravenous sting agonist, which I'll talk about today. Then immune exhaustion, we've got an HPK1, which is a kinase, hematopoietic progenitor cell kinase, which is a kinase inhibitor which allows for extra T cell action. And then we have the dual IDO-TDO inhibitor for immune suppression caused inside tumors. Very quickly, this is STING, which is the target. STING stands for stimulator of interferon genes. Almost all anti-cancer responses that are positive are associated with the generation of type 1 interferons. STING was, even though the genome is 1999 when the genome was completely revealed, Sting was not really discovered till 2012. It was not known. It's a very powerful gene, and so it's only recently come into the into the hands of drug discovery scientists. And in the last several years, there have been a lot of activity around Sting agonists and Sting antagonists, both for cancer and for inflammation. So this is a very quick sort of a cartoon. Unfortunately, I don't have a pointer here, so I can't show you very much. But if you look at the bottom, that tumor cells, and then you have these immature dendritic cells, in which when you have cell death around a tumor cell, because you have a lot of dead cells around there, cytosolic DNA is picked up. DNA is picked up by cells, and when cytosolic DNA is sensed by an immature DC, it then matures, and that process requires Sting. Because when DNA is tumor-derived DNA is seen by this by a protein called cGAS, it creates a cyclic dinucleotide called cGAMP, which binds to Sting and activates the interferon response. So that process is somehow turned down. So even though there's tumor growing, the immune system doesn't really seem to recognize it, because there is now activation of this whole pathway. This pathway is just not, it still stays dormant. But if you have, so the idea is that can you activate this pathway with a drug? Can you actually then, instead of having this process having naturally, can you activate Sting directly and start the type 1 interferon response? So what we have seen that in murine models, Sting you can just add this cGAMP directly, and you can see anti-cancer activity. And so that's what actually brought us directly to this target to start working on it. So the first generation Sting agonists from other companies that were working on it, they didn't generate very good clinical data. Even though one of the most important points is that when you injected some of them directly into the tumor, the injected tumors would really regress steeply. You'd see very major tumor regression. But because these compounds could not be given systemically, patients would have the metastatic lesions would not be accessible, because you only went into the lesion where you could inject, but the other lesions were untouched. So the shortcomings that were there of these first generation agonists was that, first of all, they were not really drug-like. The chemical structures were all nucleoside mimics or cyclic dinucleotide mimics. They didn't have the systemic tolerability that you need for treatment of all metastatic lesions. Many of them actually, interestingly, caused the degradation of Sting itself. So they would activate Sting, but then the target itself would disappear. And many of them caused T-cell death themselves. So they would, the cell type that you're trying to activate is no longer there. So obviously the therapy is not going to work. So we have here our compound, which is a allosteric Sting agonist. The point is kind of hard, but if you look at the top of this protein, where it's a CDN site for most Sting agonists is up there, that's where the cyclic dinucleotide binds. This is a cartoon of Sting, where you have Sting is a dimer, so you have these two different monomers coming together to form the binding site. And our compound actually binds on the other side, and on the distal side. And it's an allosteric site. And because of that, it overcomes shortcomings of many all the rest of the CDN site binding agonists. It generates type 1 interferons like it's supposed to, but it spares T cells. I'll show you that. It blocks a proton channel in Sting, which is responsible for hyperinflammatory responses and systemic toxicity. So therefore, it has good systemic tolerability, and you don't need tumor targeting to start it. So most of the other CDN agonists were actually delivered through tumor targeting. And recently, our tool compounds, which were actually published in 2020, were used by two independent groups in Nature and Science to show that human Sting is actually a proton channel, and that's where the toxicity comes from, and that it's the allosteric binding was demonstrated by another group in Nature in 2022. So this is the first thing, that it doesn't cause autophagy. So it has, one thing is that the pharmacology is, as I said, is distinct from other Sting agonists because of this. The other Sting agonist here is a competitor from GSK. This is the GSK's compound. And you can see, unfortunately, since I don't have this, I'll have to say, the middle, the third lane, the final, is where you have the competitor Sting agonist is the third line. So CRD3874 is our compound. You can see that wherever LC3 is a marker of autophagy, it comes up in in the case of the GSK compound, but not in our compound, where there is no autophagy doesn't occur. And this is sort of demonstrated that in the second part of that graph, where you can see that you have no killing of T cells. When you add the GSK compound, T cells die, but when you add our compound, there's no problem with T cells. They survive fine in culture. So we've done just showing you now, these are in syngeneic tumor models in immune competent mice. This is the head and neck MOC1 cell line, melanoma, pancreatic cancer, and sarcoma, and that's AML at the bottom. And all the doses are, the green dose is 30 mg per kg, the purple is 10 mg per kg, the blue is 3 mg per kg. And you can see here that if the vehicle is the red is the vehicle control. So you can see that in all of these, you can see that monotherapy, IV, you get across different tumor types, you get very strong anti-tumor reduction in these preclinical models. And same thing in AML, we should get a clean effect, extension. This is a very hard, very aggressive tumor model, C1498 cells, and they basically die, the animals die in 20 days, and you can see that there's a significant survival benefit. The last, that curve just shows that there's antigen-specific T cells, but I can't, because I don't have the marker, I'm going to skip over it. The more important question is that with cancer, will this work with other immunotherapies? And you can see that it does. There are 2 checkpoint inhibitors that we've tried. This is the one on top is PD-L1, and which in this particular tumor, which is head and neck cancer tumor, it has no effect at all. But when you combine it with a low dose of our compound, you can see that it actually starts working. So there's a synergy. So it gives you an idea that in the clinic you can try the combination. This is with CTLA-4, the one at the bottom. At CTLA-4, you can see that it's a very potent compound, but you can see that if you look at the graph, the little small graph, the inset, you can see that the number of animals that are tumor-free goes up dramatically when you combine it with a low dose of our compound. That's this. The one on the right side is the checkpoint inhibitor combination with CTLA-4 with again with our compound IV, and you can see again that there's a very large reduction in there's a good synergy with other checkpoint, and it makes sense because CTLA-4, PD-L1, all of these work in the suppressive side, and you are working over here on initiating immunity. So together there is obviously going to be synergy, and this is with an IDO inhibitor, with our own IDO inhibitor, you see synergy as well. So it combines very well with different types of immunotherapy. Now, this is just a control experiment to show you that the activity is dependent on an intact immune system. If you take animals, if you take any of these models and you add an anti-CD8 antibody, which would knock out all the T cells, then you get no activity, and that's what you see over here. The way, the activity which you see with the green line, disappears and in the presence of an antibody. You can also do this another kind of experiment. You take animals which are completely cured from the previous experiments with HNSC, the MOC1 experiments, and you re-challenge them and you see none of them get the tumor, whereas the control cohort gets the tumor. So there is immune memory, and anti-CD8 alpha actually blocks it, so it's an immune mediated action. This is just proof, a control to show that. Okay. So more interestingly, from a therapeutic, from an anti-cancer using it in patients, more just to show safety. So this was something which was very important, because when you have an immune stimulator, you can have unsafe levels of immunity, you can have cytokine release syndrome and so on. But when we tried this compound, we were able to, because of its unique pharmacology, it sort of cuts off the innate, pyroptosis and autophagy that are there in most STING agonists, seen with most STING agonists. We don't see here because it sort of separates those from just type 1 interferon generation. And so you get the type 1 interferon activity. So we did this experiment, this GLP study in monkeys in Canada, at Charles River, and we saw that there was no systemic problems even at the highest dose, at 75. And there are also no drug-related findings. The ADME properties against all the CYPs are fine, so it's got really good drug-like properties, which those are very important if you want to combine, because if you increase the levels of metabolizing enzymes, then you will have a problem with additional toxicity. So we now got ready for applying to the US FDA for clinical permission to start a phase 1 trial. And we targeted solid cancers, where we're looking at cancers which are unresponsive or refractory to immunotherapy, and we wanted to combine it with checkpoint inhibitors in phase 1A and phase 1B. And special interest was in cancers which had some evidence of a type 1 interferon signature. We're also looking at it specifically in the case of AML, because we've seen anti-cancer in the models, especially in combination with decitabine, because there's evidence that the STING pathway is activated in those patients. So the trial site is Sloan Kettering. We've started the phase 1A trial, and we're now recruiting patients there. We have seen no toxicity so far at the low doses, but as we dose escalate, maybe by the end of the year we'll get to a stage where we'll have a level where we see both activity, some activity, but right now we just have the early doses. This is the team. Dr. Govind Babu and Dr. Khatri have been part of our clinical advisory. And thank you.